name is Lauren Redpath. I am a distance education student currently living in Japan. For my semester project, I chose Botrytis cinerea in its stages in pathogenesis. Many of you may know Botrytis cinerea as the gray mold that infects strawberries before you can eat them. Strawberries are not available in Japan year-round. Therefore, I selected a fruit for the fall season, Persimmons. It is both widely popular and highly susceptible to the fungus. Botrytis cinerea is a polycyclic disease that infects either the flower or the fruit. It will then overwinter in plant debris as mycelium or sclerotia. It is produced as canidia on canidiophores, and then in the spring is wind disseminated to again infect fruit or flowers. For my project, I decided to do a time-lapse video of the disease in progress.
For my experiment, I selected host material from a farmer who used no fungicides on the tree or fruit, as I desired as natural as possible an onset for pathogenic growth. From there, I selected 3 persimmons that appeared to either have wounds or evidence of rot present. I incubated these persimmons here in this incubation chamber with a moist towelette. It took 2 days for symptoms to appear. As you see them now, it is the 2-week mark, and the scent is extremely odoriferous, smelling strongly of ethylene production. I next selected 2 fresh persimmons which had also not been exposed to fungicide. Rather than allowing natural dispersal of my inoculum, I used an inoculation needle and a pipette to induce infection. I made several wounds on the surface of 1 persimmon using the inoculation needle. I then used the pipette to transfer half a milliliter of ooze onto these wounds. On the other persimmon, I directly inoculated it by plunging this needle into the infection area of one of the incubated persimmons. Removing it, I inoculated the second persimmon. I repeated this process the same number of times as I wounded the other persimmon, so that a comparison could be examined between the 2 methods of inoculation. I then set up my time-lapse with an iPhone, as you can see here, in an environment where I could control the element of light and interaction, ensuring that no disruption or contamination befell my experiment. I am currently running a program called Lapse It to take a photo every 30 minutes, which has been running for 9 days now. Here are my inoculated persimmons. This is the one inoculated with the pipette, and this other one here is directly inoculated with the needle. You can see necrosis and browning of the tissue surrounding the infection court on both persimmons. Other factors include softening of the tissue and darkening of the fruit, especially in comparison to the fresh persimmons you see here. In addition, you can see another symptom of bacterial infection in the water-soaked margins visible here. In this image, you can see the contrast of the inside of a fresh fruit in comparison to that of a diseased fruit here. Suspicious that my disease was not the predicted Botrytis cinerea in consideration of the lack of gray mold and the presence of ooze, as well as the water-soaked margins, I decided to perform a bacterial streaming test. This test resulted in a positive sign that ultimately led to the conclusion that my subject of study was a bacteria, and not the fungal pathogen Botrytis cinerea. What I have here is a soft rot of fruit, which is often caused by Erwinia, Pectobacterium, or Pseudomonas.
The bacterial soft rot of fruit enters a host through wounds, where it can then produce enzymes to degenerate host tissue and infect. The bacteria overwinter in plant debris in the soil and will appear again in the spring when the temperatures rise. The optimal temperature is between 18 and 35 degrees Celsius, though it can tolerate warmer and cooler temperatures. The disease is polycyclic and is dispersed through vectors, such as humans, machinery and tools, and insects. It can also be spread by direct contact of an infected fruit or through a water medium after heavy rains. Though not the gray mold, Botrytis cinerea, let us continue to watch my time-lapse video displaying the symptoms of bacterial soft rot of fruits in progress.